Okay. Everyone see my full screen, right? In the
Yes.
full screen. Okay, thank you.
Yes.
Yeah,
I
thank
see.
you. Thanks. So the topic of my lecture this morning, it's about immunity to bacterial infection. The objectives of my lecture, after my lecture, we expect you to explain the general characteristics of the immunity to infections, in particular bacterial infections, against extracellular bacteria and intracellular bacteria. This means that immune to bacteria that infected and stay in cells. I will explain about innate adaptive immunity, side effects of the immune function of our immune function, that we need to clear the bacterial infection, but somehow it will cause the adverse effect or side effects. And the last is about the immune evasion, that why we still have bacterial infections. Okay. First of all, we know that bacterial infection remain a global health problem. We have troubles with bacterial infections in such as tuberculosis, in particular multi-drug resistant tuberculosis, which is a very, very serious infectious disease, because we know that Mycobacterium tuberculosis is very tough, difficult to clear from our body. And if we got multiple drug resistant, so it's very difficult to handle with this disease. And in tropical countries, we know that we have a problem about the diarrhea caused by E. coli or Salmonella typhi. Cholera is a majority of the bacterial infection that cause a massive number of deaths. And in Western country, they also have a problem with Escherichia coli, which can cause a hemolytic disease, the infectious disease caused by E. coli, and cause the severe symptoms after infect with the E. coli 0157H7. And in Thailand, we also have all of diseases that I have mentioned, and we also have the increasing number of the antibiotic resistance. So this is the overview of bacterial infections that cause the meningitis, otitis media, pneumonia, or skin infection, sinusitis, and many bacterial infection that still have the majority problem in Thailand. And in Thailand, we also have a specific disease such as melioidosis as well, in particular in northeast of Thailand. So according to bacterial infection, normally we group the infection into two groups. The first is the bacteria that get into our body, but they stay outside our cells. We call them extracellular bacteria. The other is intracellular bacteria that can infect to our cells and stay in host cells. So they use host cells as a reservoir. And intracellular bacteria is very difficult to eradicate from our body because it's hidden in our cells. So our immune response cannot recognize. And we also have the problem because these bacteria can produce some virulence factors or use some strategies in order to evade from host immune response. They may produce biofilm that can facilitate the bacterial colonization and protect the penetration of antibiotics and immune response. And they may multiply in our host cells, and they may cause a chronic infection and forming granuloma and cover with the macrophage that our T cell cannot get through to clear these bacteria. So get back to the bacterial pathogenesis. Actually, Ajahn Scarlett have mentioned already about how bacteria cause disease. So the first step, bacteria need to enter to our body via many routes, and then they got to adhere to the host using their virulence factors. After adhesion, they multiply to increase the number, because the high number of bacteria, it can resist better to our host immune response, and they may produce more virulence factors in order to attack our immune response and penetrate or invade through the deeper tissue and evade from the immune response, that finally they can cause the cell damage and cause severity of the disease. So get back to my lecture. In the essential immunology, you remember that we got innate immune response. The most important of the innate immune response is the inflammation that performed by white blood cell, particular neutrophils. If you remember this figure, this is a neutrophil that normally localized in our bloodstream. They got a number of ligand and receptor here that in normal situation, this ligand and receptor can weakly express with a low affinity state. So they can flow according to blood flow. But once we got infection, we got a macrophage that localize in the site of the bacteria get into our body. Macrophage phagocytosed and then secrete numbers of cytokines, in particular TNF and interleukin-1, and leading to the activation of vasodilation that and stimulate the expression of some ligand and molecules on the endothelium cells. So neutrophil may come to rolling next to the endothelium cell with slower speed, and because the ligand and the receptor can react high affinity and then leading to the resistance to blood flow, neutrophil monocyte can therefore move from the blood vessel to the tissue where the bacteria localize there to perform phagocytosis. So according to bacterial infection, as I said, neutrophil is a major role of the autoimmune response in order to clear the bacteria. They may eat using phagocytosis to engulf, digest of the bacteria. Neutrophil, if they got a hyperactivation by bacteria, neutrophil can release the mediators in granules. If you remember my slide of phagocytosis, inside neutrophil, they have primary and secondary granules. Their granules, they just not only release granule inside the cytoplasm. Neutrophils can excrete or secrete the mediators in granules to produce reactive oxygen species, nitric oxide species to kill bacteria. And neutrophils can form net, is a the strategy of neutrophil that can trap. They use the histone. We call them neutrophil extracellular trap. They release histone in order to trap bacteria and produce the mediators to kill bacteria extracellularly from the neutrophils. So there are three measures of the neutrophils function in order to destroy bacteria, in particular extracellular bacteria. Okay, so, and this is the flow of the innate immune response in order to eliminate bacterial cells. We use the inflammation process. We have complement that use the anaphylatoxin A, C3a, C4a, C5a in order to attract leukocytes, which is the majority of the leukocyte that can more tied to the site of infection is neutrophil. We can use C3b in order to deposit on the bacterial cell and let the phagocytic cell to adhere better to form opsonization and phagocytose. And we also have a membrane attack complex. This is a final step of complement in order to cause bacterial cell lysis. And we also have phagocytic cells, not just only neutrophils. We also have dendritic cells, macrophage, that can ingest and release some cytokines, interleukin 12 to activate NK cells, interleukin 6 in order to activate liver to produce acute phase protein and cause fever, and IL-8, IL-1 TNF alpha in order to cause vasodilation to increase vascular permeability and let the white blood cell extraction from the blood vessel to perform phagocytosis to release mediators to clear the pathogens. We also have the mucous membrane here that can produce the mucus that interfere the adhesion step of the bacterial pathogenesis. We have numbers of innate immunity acids, which is the not can create very low pH that not suitable for the bacterial growth. Lysozyme is an enzyme that can cause the bacterial leakage because lysozyme degrade peptidoglycan of gram-positive bacteria. There are also many digestive enzymes, protease, lipase, DNase, RNase. Defensin and catalycidin is the cationic protein that have a positive charge that can anchor and cause pore of the bacterial cell. Okay, if you remember that, and we also have the commensal microbes with normal flora. Okay, and okay, this is a phagocytosis here and inflammation here. I think you quite familiar now. If you study by yourself, step by step, you will understand how can innate immune defense against pathogens. And this is the how dendritic cell capture antigen and then carry the phagocytose and carry antigen to prime naive T lymphocyte that localize in lymph node in order to trigger the signal to stimulate the adaptive immunity to let T cell know that we got infection. And this is a step of phagocytosis, right? And then we got a nitric oxide here, or reactive oxygen species, in particular hydrogen peroxide to kill microorganisms. We also have macrophage that can not only phagocytose but release lots of cytokines to stimulate our immune response. Okay, and we also have inflammasome, and finally we got a cytokine that can cause acute inflammation. We have toll-like receptor that recognize extracellular bacteria, because some of toll-like receptors localized on the surface of our host cells like this. So you can see toll-like receptor 1 and 2. Normally toll-like receptor locate on the cell surface in dimer. Toll-like receptor 1 and 2 can recognize bacterial lipopeptide. Toll-like receptor 2 recognize peptidoglycan. Toll-like receptor 4 recognize LPS. Toll-like receptor 5 recognize fractalin. And if toll-like receptor 2 stay with toll-like receptor 6, it can recognize bacterial lipopeptide. So this is how our white blood cell recognize bacterial to differentiate that this is the pathogen. But if the bacteria is intracellular bacteria, we also have toll-like receptor and other the signaling pattern recognition receptor to recognize intracellular bacteria as well. Okay, and NK cell also play a role in the bacterial infection, because NK cell can recognize the infected cells and release, not only kill infected cell, but also release interferon gamma to help macrophage. Okay, NKT cell, if you remember, NKT cell, they got a receptor that is the invariant recognition to recognize lipid and glycolipid. That is very important to detect bacterial cell by innate immune response. After recognition, NKT cell can release cytokines to stimulate the other cells. And we also have gamma delta T cell, which can stimulate many immune response. Okay, antigen presentation is very important to link the innate immunity and stimulate adaptive immunity. So we got dendritic cell, macrophage, and B cell that can initiate, dendritic cell initiate the adaptive by trigger naive T cell to have the clonal expansion and differentiation from naive T lymphocyte to effector T cell. That effector T cell mean the T cell that ready to work, that were primed with a specific antigen. And macrophage trigger T cell that localize near the macrophage, because the macrophage can localize in numbers of our organ and tissue, so they can present antigen to effector T cell nearby. And B cell can act as antigen presenting cells to get the signal from T cell, and then B cell will differentiate from B cell to plasma cell and produce antibody. Okay, and this slide shows the various effector T cells that can defend against bacterial infection. T helper 1 is the T cell that can secrete interferon gamma to tell macrophage, to help macrophage. So macrophage that engulf pathogen can have the active reaction to kill intracellular pathogen that they engulf. T helper 2 play a role in the eosinophil function and mast cell function. That majority of T helper 2 is a host defense against helminthic infection. The most important of T helper that play important role to clear bacteria is T helper 17. They name T helper 17 according to interleukin 17 that these T cells secrete. And T helper 17 also secrete interleukin 22 as well. So the target of T helper 17 is neutrophils, because this cytokine can recruit the neutrophil to the site of infection and defense against extracellular bacteria or fungi. Okay. And T follicular cell here can play a role in B cell function to initiate the antibody production. So these are, okay, I'm going to the how immune defense against extracellular bacteria. Extracellular bacteria is bacteria that stay outside our cells. They may stay in our extracellular matrix or in any tissue. The majority of the main problem of the extracellular bacteria infections caused by Staphylococcus aureus, Streptococcus pyogenes, and E. coli, VBO, and Clostridium occurring Bacterium diphtheriae. So how could our immune defense against extracellular bacteria? So once we got infection with extracellular bacteria, we have our tissue, our mucous membrane, and we got enzyme proteins and to defend it. But these bacteria can evade our tissue to the deeper tissue and cause disease, because they can produce numbers of virulence factors. The most important virulence factors are toxin. They are exotoxin and endotoxin that Ajahn Scarlett have lectured already. Exotoxin is they are proteins secreted by gram-positive or gram-negative bacteria. They may enterotoxin that cause the disease of our GI tract. Neurotoxin, cytotoxin, and leukocidin or hemolysin is named after their function. Okay, and bacteria can produce endotoxin, which is the LPS. This is a heat stable antigen. It's not sensitive to heat, but exotoxin is protein, is sensitive to heat, but endotoxin is resist to heat. So endotoxin can cause numbers of the adverse effect in our body, such as they can stimulate the production of interleukin-1, secretion of histamine, tumor necrosis factor, and also number of mediators. You can see that. So if we got a heavy infection of bacteria, in particular gram-negative bacteria, and they produce numbers of LPS, so you may get chills, high fever, leukopenia, and endotoxic shock. Okay, and in the pathogenesis, if we got numbers or high level of endotoxin, you may have a increase the level of cytokine, TNF alpha, IL-6, IL-1, and these HMG1, IL-8, pellet activating factor, complement, kinin, and histamines. Normally, these mediators produced in order to cause inflammation, to clear bacteria. But if you got a heavy infection, you may have a high active of these cytokines, and you got inflammation too much, you got nitric oxide too much. Remember that when you got inflammation, these mediators can cause vasodilation. So not only white blood cell get through the blood vessel to the tissue, they are also the fluid from your blood vessel leak from your blood vessel. So you have the imbalance you got hypotension because no fluid in your bloodstream. So your blood will not reach the vital organ. So you got a organ hypoperfusion that can cause the lung, heart, kidney, liver dysfunction and cause organ failure and cause death finally. So the higher cytokine level that out of control of our body, we call them cytokine storm. That is a very important. Even in COVID-19 patients, they got infection and their immune stimulated, and they got a high level of cytokine, leading to cytokine storm and cause the severity of the patients, and then may cause organ failure and death. Okay. Another virulence factor is super antigen. Ajahn Skarat also talked already. Super antigen is the antigen that can bind to the MSC of the T cell receptor without any antigen here, or it may be any antigen. So if we got a super antigen, your immune response will stimulate out of control, too much activation. So it can cause severity of the disease as well. Okay. And so that's the first immune response, in particular innate immune response. The next is about humoral defense. We know that humoral immune response is the soluble immune response, which are antibody. So when we get infection, normally our immune response, we produce antibodies. Antibodies can neutralize microorganisms. Antibody can bind to the bacteria and phagocytic cell that has receptor to Fc of antibody can capture this antibody that we call opsonization and facilitate phagocytosis. Antibody that bound on the infected cells, NK cell have Fc receptor. NK cell can bind and secrete perforin and granzyme in order to destroy infected cells. Complement, if we got antibody bound to the antigen, this is immune complex. Therefore, complement, which pathway of complement, if we got antigen antibody reaction, which pathway of complement are activated? Okay? Classical, right? It's classical pathway. So after classical pathway activation, you will get inflammation because you got C5a, C4a, C3a, right? And you got opsonization, we got massive amount of C3b, and we got a membrane attack complex. So this is the answer, right? After you got activate classical pathway, so you got inflammation, opsonization, and also the membrane attack complex. Yeah. So adaptive immune response against extracellular bacteria. We got antibody to neutralize, opsonize, phagocytose, inflammation, and lysis. And for the T cell, what they have done, in bacterial infection, once bacteria get into our body, we got phagocytic cells. Phagocytic cell phagocytose bacteria and then digest and present antigen with MST class 2 to send the signal to helper T cells, CD positive. Once CD positive helper T cell recognize that we got infection, so T cell will release interleukin, in particular interleukin 17, which is best strategy to defend against bacterial infection. We got TNF alpha and other cytokines in order to induce inflammation. T helper also release interferon gamma that facilitate the phagocytosis mechanism of phagocytic cell to kill bacteria. And T helper cell can release number of cytokines to facilitate the differentiation and proliferation of B cell to develop to plasma cell and produce antibody. However, even we know that we got very excellent immune response, but anyway, some bacteria have evasion strategies to avoid the function of our immune response. I'll give you an example of some bacteria that can avoid antibody function, such as Nyseria gonorrhea. Nyseria gonorrhea is gram-negative bacteria. They can change their amino acid on the cell surface. Therefore, even we got infected Nyseria gonorrhea before we got antibody, we got immune response against gonorrhea. But when they change amino acid, so we cannot recognize this new model of this Nyseria gonorrhea. Haemophilus influenza can express the IgA specific protease. We know that Haemophilus influenzae infect to our respiratory tract. Normally in our respiratory tract, we got IgA, secreted IgA in order to defend against bacterial infection via the mucus. But these bacteria can produce IgA protease to degrade IgA, so they can survive because there are no IgA enough to kill them. And some bacteria such as E. coli, they can produce sialic acid on their LPS, because sialic acid is our host molecule. We got sialic acid, so our immune response cannot recognize this bacteria is pathogen or is foreign body because they have a similar amino acid, not amino acid, they have similar sialic acid as we have, so they can evade from our immune response. Some bacteria can perform the membrane vesicle like a decoy to cheating our immune response. So our antibody detect, oh, this is antigen. So antibody react to this, but cannot react to the main organism. So they can evade from our immune response. And this is some bacteria can change their antigen. And some bacteria can avoid phagocytosis. As I explained, phagocytosis is very important mechanism in order to engulf, digest, present antigen. But some bacteria, some pathogen can avoid our phagocytosis mechanism. The first, some bacteria can produce capsule to cover their cell. So we know that our phagocytic cell have PRR to recognize PAM, but bacteria produce capsule to cover the PAMs. So our phagocytic cells cannot see the PAMs. They may change the charge of the surface that avoid from phagocytosis as well, or the change by produce capsule to cover bacterial cell can interfere C3b localization. So there are no C3b, so we have no C3b bind to the cell surface, so we cannot recognize by opsonization and phagocytosis. Some bacteria can enter to non-phagocytic cell. Bacteria can infect to epithelium cell of fibroblasts and stay inside, hidden inside our cell. So our antibody cannot detect because they live inside host cells, and they got type 3 secretory system in order to control the rearrangement of our cytoskeleton to uptake bacteria into host cell and hidden in our host cells. Bacteria also produce some virulence factors to interfere phagosome-lysosome fusion. If you remember phagocytosis, once bacteria engulf, they stay in phagosome and then fuse with lysosome, because in lysosome there are numbers of agents that can kill bacteria. But some bacteria can interfere the fusion between phagosome and lysosome. Or this one, normally they get into our phagosome here. Lysosome should fuse with this phagosome, but they evade or block the fusion between lysosome and phagosome and stay and replicate inside the phagosome like this. And they may escape from phagosome by producing some enzyme, so bacteria can get off the phagosome and stay in cytoplasm of our host cells. Some bacteria can avoid complement because complement can kill bacteria, cause opsonization, phagocytosis, cause the inflammation and membrane attack complex. Treponema pallidum that cause syphilis can avoid C3b deposition by using their outer membrane, and they may produce longer LPS to prevent membrane attack complex. And not only E. coli, streptococci group B also have the salicylic acid that can mimic our host cell and also degrade the C3b and block the alternative pathway. And Salmonella, gonococci, meningococci can interfere the complement cascade. If you see in this figure, it's more easier. So this is supposed to be classical pathway that we got antibody by to the bacteria. So Staph aureus can bind IgA and block the binding between antibody to the C1. C1 is the complement component. Some bacteria can interfere the stabilizing factor here or inhibit C3 convertase and inhibit the C5a production, because C5a is the Anaphylaxis toxin that can cause inflammation. So many bacteria have their strategies to avoid the killing mechanism by complement. Okay, so that's all of the extracellular bacteria. The main function of our innate that can destroy extracellular bacteria is the antibody, complement, and phagocytic cell. Okay, let's move to the next group of bacterial infection, is the bacteria that are intracellular bacteria. How our immunity can defend against intracellular bacteria. So this is the disease mechanism of the intracellular bacteria. They get into our body, they can endocytose in many cells, and they replicate in our host cells and then can spread all over our body, in particular if that bacteria can infect into macrophage. They may use macrophage like a reservoir, or some scientists normally call it Trojan horse. If you remember Trojan horse in the movie Troy,ใช่ไหม? Troy นะ Trojan horse is the reservoir that can carry the pathogens and spread throughout our body, disseminate into our body. Okay? So again, we know that we got innate and adaptive immunity. In order to control our immune response, we got neutrophil, macrophage, T cell and B cell to control the bacterial infection. Okay. The immune effector mechanism in order to control intracellular bacteria, we got neutrophil, macrophage, and antimicrobial peptide and inflammation cytokines, and NK cell, gamma delta T cell is very important to control intracellular bacteria, because once bacteria can get into our host cells, antibody hardly recognize the pathogen. But we got NK cell. If you remember, NK cell, we use the activating receptor and inhibitory receptor in order to recognize this is an infected cell or healthy cells. So the infected cell normally got a different expression of MHC class 1. So NK cell know they can produce perforin and granzyme in order to cause apoptosis of infected cells. And we also have the gamma delta T cell that can produce mediators to cause cell lysis. Okay. Another important factor to control intracellular bacteria is cytotoxic T cell or CD8 positive T cells. These cells are supposed to be the infected cells. After infected, the cells degrade antigen and present antigen through the MHC class 1. Every nucleated cell got MHC class 1. So if our cells infected with antigen, our cell process antigen and present antigen on the cell surface via the MHC class 1 in order to tell the other cell that we got infected. So this infected cell can be recognized by CD8 positive cytotoxic T lymphocyte. After recognition, this is the infected cell. CD8 positive release perforin. Perforin is a protein that cause a pore on the cell surface and then let granzyme into the infected cell. Granzyme means the enzyme in the granules that contain many type of the enzymes inside the granules. So we call them granzymes. Inside granzymes, they can trigger apoptosis and cause cell death of the target cells in order to clear the antigen. So the babbling of the cells of apoptotic body will be cleared by macrophage to clean and eliminate the pathogen. Cytotoxic T cell can also activate cell killing via the fast ligand as well and cause apoptosis. Okay. T helper 4 or T helper or CD4 positive cell also play a role in intracellular bacterial infection. So let's have a look step by step. You got phagocytic cell. This is supposed to be dendritic cell. Dendritic cell engulf microorganism and present antigen to MHC class 2 to initiate naive T cell in lymph node. Naive T cell, after activated, they proliferate and mature into the effector T lymphocyte. And this cell can be differentiated and maturation by numbers of cytokines secreted by macrophage and NK cell. So this can be function and can be differentiated into T helper 1, T helper 2, or T helper 17. So in classical macrophage activation, in order to clear bacterial infection, we got naive T cell to produce cytokine and initiate T to T helper 1 or T17 and facilitate the activation of macrophage that engulf pathogen here, but somehow it may not clear properly. So when they got interferon gamma, this macrophage can easily clear the antigen, is enhanced microbial killing. And this is also the phagocytosis that ingest microorganism in vesicle. So when we got infected, you can see macrophage send the signal to T helper 1 and T helper 17 to secrete the cytokine to facilitate the bacterial killing, as well as activate the inflammation in order to kill microorganisms, kill bacteria. And there are also the cooperation between CD4 positive and CD8 positive. We got phagocytosis bacteria in macrophage here. Macrophage send a signal to T helper. T helper secrete interferon gamma to help macrophage to kill bacteria in phagolysosome. And macrophage also send the signal, because in macrophage we have MSC class 1 and class 2. MSC class 2 with antigen trigger T helper. MSC class 1 with antigen also send a signal to cytotoxic T cell. So cytotoxic T cell will release perforin and granzyme to kill infected cells. So in the immune response, they work like orchestra. Every cells help to each other, and spontaneously they can synchronize the function. They can act in the same time. It depends that what mediators we have or what we got at the effector cells. Okay. For intracellular bacterial infection, the humoral defense can control the infection not only using the antibody bind to free and bacteria, because now bacteria is in our cells. So how humoral defense help to eliminate bacterial cells, so they can help to bind to the cell and opsonization here, and leading to the phagocytosis, or even the cell infected with bacteria and have antibody that can bind to the antigen. Phagocytic cell can digest as well. And NK cell can bind to the FC of immunoglobulin here and release perforin and granzyme to cause apoptosis of the infected cells. The sixth mechanism of our immune response to defense against intracellular bacteria, we call them granuloma formation. Granuloma formation is the interaction of our cells in order to, if you look at on the right hand side first, this is a macrophage and this is the bacteria. Normally macrophage can digest and cause bacterial cell lysis or cell death, but some bacteria can resist and stay in macrophage like this. So macrophage carry these bacteria, but we got numbers of cytokines to facilitate the macrophage to kill bacteria and cause the death of the bacteria inside the macrophage. But some bacteria, such as Mycobacterium tuberculosis can resist to these two reactions. So macrophage will carry the bacteria inside, and the cytokine secreted by infected macrophage can recruit T cell to the site of infection in order to clear the infected cells like this, right? But if we cannot clear the infected cell inside macrophage, our body will gather all T cell and macrophage and form granuloma to limited the area of macrophage to stay that site, not spread to the other areas. Unfortunately, if we cannot clear off this granuloma formation, and someday there are some disturbed accident that can cause the leakage of bacteria, it can cause a major problem to spread the bacteria from the macrophage throughout our body. Okay, so in summarization, I think to be summarization for the our immune response defense against bacterial infection, we got neutrophil, NK cell, macrophage, and dendritic cell in order to clear the antigen and send a signal to the adaptive immunity. We got a cytotoxic T cell to defense against the infection by perforin and granzyme. We got a signal from antigen presenting cell to trigger CD positive and proliferate CD positive to and differentiation to mature T cells, and secrete and cytokine to stimulate B cell to plasma cell and produce immunoglobulin. Immunoglobulin also attack to the bacterial cell and function along with the innate immunity, such as opsonization and complement activation. And the evasion mechanism of intracellular bacteria to avoid phagosome or phagocytosis is also a major problem, such as Mycobacterium leprae can infect non-phagocytic cells such as Schwann cells. Listeria monocytogenes can escape from phagosome because they have the enzyme listerio-lysine to lyse the wall of the phagosome and escape from phagosome to the cytoplasm. Mycobacterium as well, and Mycobacterium tuberculosis, right, can inhibit the fusion between phagosome and lysosome. Mycobacterium tuberculosis can produce ammonium ion. If you remember in phagocytosis, inside phagolysosome is very acidic pH, but some bacteria can produce ammonia in order to neutralize the pH that can manipulate environmental for the bacterial survival. And Salmonella can reduce the production of NADPH and inhibit the reactive oxygen species and reactive nitrogen species. Bacteria pseudomonas as well can facilitate, they can interfere the phagocytosis to interfere the production of ROS and nitrogen species. Okay, I think if I finish this slide, I will take a break for 5 minutes. Okay. The next of the strategy of bacteria to avoid antibodies. Intracellular bacteria is really clever. They try to get into our cells and stay inside our cells. But once they stay inside our cells, they are not stay like that. They try to escape and infect to the neighbor cells. There are numbers of studies in Listeria monocytogenes, Bacterial Pseudomallei, Salmonella, Shigella, and Rickettsia. They have a very similar strategies in order to avoid antibody, because if bacteria stay inside cells, antibody cannot recognize. But if they would like to infect the other cells, how would they do? There are evidence demonstrated by scientists. If you look at this figure on the right top corner, the bacteria, this is Listeria, phagocytose into the phagocytic cell like this and stay inside. Okay, they live in the phagosome, and they got enzyme to lyse the phagosome wall and get off into cytoplasm. When they stay in our cytoplasm cells, they would like to infect the next. They produce protein to regulate our actin filament to push them like a เขาเรียกกระสวยอ่ะอาจารย์จำชื่อภาษาอังกฤษไม่ได้ like a shuttle, like a shuttle. So they manipulate the actin filament and protrude into the next cell without exposed to extracellular cell and stay in the next cell. So this is the same explanation. They got actin filament rearrangement and direct there to project into the next cell and stay in the cytoplasm of the next cells. And this is the Bacterial Pseudomallei stain with the red cells here. The green is the color that stain actin filament. You can see these bacteria control our actin filament and control them to project into the next cell nearby. Okay. Okay. So the next slide is how bacteria avoid T cell. But I think I better take a break for 5 minutes. Okay. And then you can get back. If you're back, please turn on your camera. Okay, to check that everyone is alert, still alert. How are you? How are you now? How do you feel? This lecture, I got plenty time to talk. It's not last time that too much content.
I feel my brain is broken now.
No. Stay with me. I know. But you can do your best now. You may the study style in graduate study is different from undergrad, totally different. Yeah. No? There are more information, there are more content, and you need to study by yourself. And I know online teaching is a big barrier to learn. You feel very exhausted than normal. So I hope everything is getting better now. Okay, let's continue. I just checked that I got couples of slides left, and we may have time to discuss or ask questions, not only this topic, maybe my previous lecture. Okay. The third mechanism of the intracellular bacteria to avoid our immune response is they may down-regulate the surface marker, in particular MHC class 1, class 2, and CD1. Mycobacterium tuberculosis is intracellular bacteria and mainly and deeply studied by researchers. So we know quite a lot of these bacteria. Therefore, we can use Mycobacterium tuberculosis as a model to explain the mechanism how intracellular bacteria avoid the killing by T cells. We know the normal step after bacteria ingest by macrophage. We got gamma delta T cell here, we got T cell, we got NK T cells in order to try to gather to get rid of these bacteria. And we know that we got MSC class 1 to present antigen to trigger CD8 to produce perforin and granzyme to trigger T helper to produce numbers of cytokines here to activate T helper 17, T helper 1, T helper 2, and so on. But Mycobacterium is very clever. They try to survive. After they ingest by macrophage and get into the phagosome, normally phagosome and lysosome can fuse, but MTB can block the fusion between this lysosome and phagosome. So they can stay in phagosome freely and multiply in phagosome. And they can also interfere the expression of MSC class 1 because MSC class 1 can send a signal to tell the other cells. So they block the expression of MSC class 1 and MSC class 2 as well as a CD1. So other cells of adaptive immunity cannot get the signal that we got infection. So MTB can live in the infected cells and spread to the vital organ and causes severe of the tuberculosis. Okay. And finally, I just would like to summarize that once we got infection, we got innate immune response, we got adaptive immune response, and we got a memory cell into recognize the previous infection. So if you look at this figure on the right hand side, when we got infection after innate immune response, this is the innate adaptive immune response. We got the trigger of naive T cell or naive B cell to be activated and proliferate into effector cell, B cell, plasma cell, produce immunoglobulin or T cell to be effector T cell, and we got a memory cells here when we infected again. So we shorten the time to produce a secondary immune response to produce higher immunoglobulin and more number of T cell, and also we got a memory cells. So this is the summarization of the our immune response. But anyway, in bacterial infection, both innate and adaptive can function, but bacterial cells have the virulence factor in order to avoid our immunity. So we can get severe disease if we cannot control, or we may get an adverse effect, in particular like cytokine storm. Okay. Oh, this is my last slide. This is the reference, and this cellular and molecular immunology, you can read ebook via the clinical key in the our library website, and there are numbers of textbook, which is, I think it's more easily to understand, because cellular and molecular immunology is quite complicated when they explain using the word to explain. But if you start with the author's book before you go to cellular and molecular immunology, it would be better for you. Okay. So that's all of my talk. And if you have any question ฉันจะออกในบ่า okay. And actually I got numbers of video that may facilitate your understanding, but I think I better post it in the e-learning that you can access by yourself, okay? But better than I show you here because maybe waste your time. Okay, do you have any questions?
Can you please explain the net release again of the neutrophil?
On the netosis, right? The netosis,
Yeah.
right? Netosis is when we got heavy infection or high number or high virulence factors of bacteria. Normally, neutrophils cause phagocytosis, right? Engulf, digest, right? But neutrophil can release granule to destroy bacteria. But netosis is the mechanism that neutrophil release histone and their chromatin in order like a trap is the like a ตะข่ายภาษาอังกฤษเขาเรียกอะไรอ่ะเดี๋ยวนะอาจารย์นึกคำไม่ออก net ครับ like a net ขอบคุณค่ะ like a net in order to produce from the cell นะ and trap bacteria immobilize bacteria during the immobilize their granules also release the mediator to kill bacteria as well. So this is the new.
So in that release, they release some kind of histone, the protein that bind to DNA. Is it right?
They release the DNA of neutrophil because it's a chromatin, no? They're like a net, that chromatin and chromosome with histone. No? You know that chromosome composed of the DNA that wiring over the histone, right? They protrude this chromatin to trap the bacteria, because if we got a highly virulent bacteria, phagocytosis is not enough. They got another mechanism to control the bacterial number, not only phagocytosis. I think I may have animation, but I don't know where is it. in my lab, we also perform netosis against Bacterial pseudomyeliae, and it is very interesting that there are in deeply knowledge, there are many formation of nets. I think you may get more information in seminar because Wong Sakon, the second year student, he is going to do the netosis as well. So he may give a talk about netosis that may give you more information how neutrophil use netosis to control bacterial infection. It's a very interesting topic.
And another thing is about cytokine storm. How can we know that if the person is got infected and there's an cytokine storm in that person?
Right. Normally, when the doctor take care of the patients, cytokine storm is the situation that doctor don't want to occur. Once they then observe the patients, they got fever, right? If the fever is too much, and they check the level of interferon gamma in the blood vessel. Interferon gamma, we know that this is a magic cytokine that facilitate the inflammation. But if the patient have a very high level of interferon gamma, it's very dangerous because too much reaction of immune is not good. So the patient may have some character. They got a hypotension because when we have a severe inflammation, you got a leakage of the fluid from the blood vessel. You got vasodilation, hypotension, and the temperature will change, the cytokine level is changed, so it will cause a cytokine storm. Normally, they may manage with the steroid in order to reduce the inflammation reaction, but managing using steroid is also double sword. Is also dangerous to balance between the interaction of immune, because if you have too much steroid, you may get the infection. So the doctor know how the patient can get they can observe when the patient get cytokine storm, but it's very difficult to manage.
So that's why in COVID, patient they use like glucocorticoid, right?
Yeah.
Okay.
They have to balance. Balance everything, because once you got imbalance in your patient, it's hardly to control. There are many factors.
I have one thing. What is cytopathic effect?
Cytopathic effect. Cyto is mean cell. Pathic is pathology. Effect is mean it happened. It means that once we got infection, the pathogen can cause pathogenesis and cause the change of our cell. So the cytopathic effect is the character of the cell that have changed according to the pathogenesis, such as if cell infected with virus, you can see cytopathic effect. You may see the multinucleated giant cells, you see the clumping of the cells or the lysis, something. It's the group of action, group of the effect, not action, group of effect that caused by the pathogenesis of pathogen that occur in cells.
So the bacteria also cause the cyto effect too?
Yeah, it can cause. For example, because I work with Bacterial pseudomallei. Bacterial pseudomallei is intracellular bacteria. They can cause multinucleated giant cells like virus infection. It can cause because if you remember when the bacteria can protrude using actin filament by Listeria, Bacteria also perform like that. And then they can protrude to the nearby cells and cause the multinucleated giant cells. It means that bacteria can fuse our cells together. Many cells have the same membrane, but actually they fuse many cells together. So we call them multinucleated giant cell. Firstly, we found in only virus infection, but numbers of bacteria can perform like this. Listeria, Bacteria, I'm not Listeria, yes. Bacteria, yes. I'm not quite sure about Salmonella and Shigella. How about Innate on Wednesday? Are you okay? Have you got take a look again to my video? But if you have any problem, you may email me. And I'm thinking about how to access your knowledge. I will arrange the test, right? Maybe by the end of the course. It's okay for you? Right? I think, do you have the head of your group that I can control you, or you have any contact that I can contact to you to arrange the exam date? How long you need to read before you take exam?
like 1 year.
1 year is
Yes,
too long.
1 year.
1 year too long. No,
I have
1 year
question.
you know, 1 year that you 1 year that you forgot everything I talked today,รู้ไหม? It should be a week after the course นะ I don't know the other lecture, the other อาจารย์ what did they manage? Some อาจารย์ take the score, right? ใช่ไหมคะ บางอาจารย์เก็บคะแนนเลยใช่ไหมคะ Some อาจารย์ test you no
ยังครับ
เหรอ อาจารย์น่ะสงสารพวกคุณ we know that if I take the exam in the lecture นะ you will go to okay I'm happy if you ask me any question, but I think if we ask and answer in the class, it's better to normalize your knowledge ใช่ไหมคะ Okay, we got about 20 minutes before we leave the class.
Yes.
Okay, any question, please? So we have กี่คนเนี่ย10. We have 10 now, right? Stay with us นะคะ don't leave anywhere นะคะ Any?
I have question.
Yes.
Can you explain me more for the multi-drug resistant? Only tuberculosis has a multi-drug resistance that you mentioned me in the slide.
You mean the mechanism?
Yes.
Mechanism. For the drug resistant cause in bacteria, I think Ajahn Tritima will give a next lecture, maybe, right? But overall, drug resistance can caused by many mechanisms. Some bacteria produce enzyme to degrade. Some bacteria got the efflux protein to pump the drug off their cells. Some bacteria can produce biofilm to block the penetration of the antibiotics. Actually, there are many mechanisms, and some bacteria can modulate their gene to produce some protein to interact with the antibiotics. In my field, I know that Bacteria pseudomallei again, they produce extracellular DNA, which is the negative charge to neutralize the positive charge of some antibiotics. So I think bacteria is try to survive. They use many things to block the harm substance to kill them.
And one more question for extracellular and intracellular has a different for the extracellular has only one normal pathogen, and intracellular has a like pathogen, very high pathogen. I mean the like Bacterial Pathogenesis or Nyseria Gonorrhea is intracellular, for example.
You mean that, if I not correct me, you mean that the bacteria can perform intra or extracellular location at the same time? No, normally some bacteria, extracellular bacteria, they love to stay outside cells, right? So, but intracellular bacteria, they prefer to get into host cells. But in intracellular bacteria, some stage of the infection, they may spread from the cell and expose to outside. So our antibody may detect them, but mainly they prefer to stay inside cells because they need something for their growth, or they may have receptor to bind to their PAMs and get them into the cells. It's the nature of the bacteria to prefer stay inside, because when they get into host cell, you know, inside our cell may have very limited resource of nutrients, but they may use some strategies to capture or get advantage while they stay inside. So we normally group them as intra or extra, but sometimes extra may be endocytosed or phagocytosed, but they not stay in cell properly. But if they are intracellular bacteria, they try to get into host cell to stay inside host cells for their survival. Any question? If not, okay, any?
Not now, maybe.
Okay.
Maybe I will please look your slide again. Maybe
Okay.
I have question.
Okay. I love to. Okay, I love to take any questions now, and I try to choose my clip video that I download from YouTube or any resource, but it's may help you to understand more. But if you have question, email me, and when I answer, I try to answer and post it in the e-learning. It's okay, because you will learn yourself now, because you change your learning habit now. You are not graduate student, and you are not undergrad student now, you are graduate students. So thank you for your attention to my talk, and hope you are adapt yourself and be a happy and success student นะคะ Okay, see you.
See you.
Bye bye.